Have you ever really, truly looked at a blood bag? It seems so simple, right? Just a plastic pouch. But inside that bag is a century of incredible science, a whole story of innovation designed to solve one massive problem. So let's dive into the amazing journey of how we figured out how to keep blood alive outside the human body. This was the fundamental question. The second that blood leaves our veins, a biological clock starts ticking, loud. Its first, most immediate instinct is to basically self-destruct. So those early medical pioneers, they weren't just fighting against time. They were fighting against the very nature of blood itself. So before anyone could even dream about long-term storage, they had to deal with the immediate threat, clotting. Blood is designed to plug leaks, right? The moment it hits the open air, it starts turning into a solid mass. This was the first giant wall they had to get over for any transfusion to even be possible. And the first attempts, wow, they were frantic and honestly pretty grim. You had doctors trying these direct side-by-side transfusions, literally racing to get the blood from a donor to a patient. They tried whipping the blood to physically pull out the clotting fibers. They even tried using special wax-coated flasks, hoping it would slow things down. But as you can imagine, none of this was reliable or scalable or particularly safe. They desperately needed a real scientific breakthrough. And then, in the early 20th century, it happened. That breakthrough finally arrived. It was a simple chemical discovery, but one that would completely tame the clotting process and turn the idea of a blood bank from a fantasy into a real, tangible possibility. So the hero of the first part of our story is sodium citrate. An anticoagulant is just what it sounds like. It stops blood from coagulating, or clotting. And sodium citrate is a genius at it. It works by binding with the calcium in the blood, and that calcium is a key trigger for the whole clotting process. So it essentially just switches the whole thing off. Just like that. The frantic race against the clot was finally over. And what's so cool is how quickly one innovation sparked the next. In 1914, you have sodium citrate solving the clotting problem. Then, just 2 years later, right in the middle of World War I, Rouse and Turner have another idea. What if we add some sugar, some glucose, to the mix? This simple step gave the blood cells a food source and bam, we have the very first short-term storage of blood. Okay, so this is a crucial point. Solving clotting, that was only half the battle. Now scientists were face-to-face with an even more complex challenge. Great, the blood isn't a solid lump anymore, but the red blood cells themselves are slowly dying in the bag. So how in the world do you keep billions of individual living cells alive and functional? Well, to solve that, we have to understand what a red blood cell actually needs to survive. In your body, it's got it made. A constant supply of food, a perfectly stable environment. But in a bag, it's on its own, and its internal systems start to break down pretty fast. Let's think of ATP as the cell's personal, rechargeable battery pack. A red blood cell actually breaks down glucose to generate this ATP, and that ATP powers everything, especially keeping its membrane flexible and strong. When the ATP levels start to drop, the battery dies, and when the battery dies, the cell fails. Now, here's the other tricky part. Just carrying oxygen isn't enough. The cell has to be able to let it go when it gets to where it's needed. That's the job of a molecule called 2,3-DPG. You can think of it like the key that unlocks oxygen from the hemoglobin. If you don't have enough 2,3-DPG, the red blood cells become super stingy with their oxygen, which makes a transfusion way less effective for the patient. So scientists started to map out these key problems, and they gave it a name, the storage lesion. Basically, it's a three-pronged attack. The cells run out of power as their ATP drops, their outer walls get stiff and damaged so they can't squeeze through tiny capillaries anymore, and they stop being able to deliver oxygen because their 2,3-DPG levels plummet. This was the new enemy they had to defeat. And what came next was this decades-long race against cellular decay. And it was a race fought entirely with chemistry. Scientists began cooking up these new preservative solutions, each one designed to specifically target the problems of the storage lesion. And you can see the progress just clicking along. In 1943, during World War II, you get ACD solution. That's followed in 57 by CPD solution, which added a really crucial ingredient. And then, in 1968, CPDA1 comes along and adds the final piece to the puzzle, which led to a massive leap forward. You can really see how the recipe evolved. ACD was a pretty simple mix of citrate and dextrose. Then, with CPD, they added phosphate. That helped keep the pH stable and did a much better job of preserving those vital 2,3-DPG levels. Then came CPDA1, where they added a tiny bit of adenine. Now, adenine is a direct building block for ATP, so it helped the cells keep their batteries charged for much, much longer. So for years, with both ACD and then CPD, the gold standard for storing blood was 21 days, 3 weeks. I mean, that was a huge achievement, but if you're a blood bank trying to manage supply, every single extra day is critical. And that is why the addition of adenine in CPDA1 was such a revolution. By giving the cells the raw materials to make more ATP, it boosted the shelf life from 21 days all the way to 35 days, a full 5 weeks. This was an absolute game changer for blood banking logistics all over the world. But the innovation, of course, didn't stop there. The next huge leap forward didn't come from changing that first preservative, but from adding something extra after the blood was collected. And this brings us right up to the science inside the modern blood bag. Okay, so here's a really key distinction to understand. The bag that the blood is first drawn into has that anticoagulant, like CPD. But today, we usually separate blood into its components. We take out the nutrient-rich plasma for other uses, which leaves the red cells behind. So to make up for that, we add a special additive solution, like SAGM, which is like a superfood cocktail of serine, adenine, glucose, and mannitol. It gives the cells an extra boost of everything they need to survive even longer. And these additive solutions, that's what gets us to the standard we have today, a maximum shelf life of 42 days, 6 full weeks for refrigerated red blood cells. When you think about it, going from a few frantic moments to 6 weeks, the progress is just astounding. But all that amazing chemistry was only half the story. The container itself was a huge problem. Before the mid-70s, blood was stored in these heavy, fragile glass bottles that had to be washed and sterilized perfectly. The switch to disposable plastic bags was honestly just as revolutionary as the chemicals inside them. Oh, the benefits were enormous. The flexible bags made it super easy to spin the blood down and separate it out into plasma, red cells, platelets. So one donation could now treat multiple patients. Being disposable, they slashed the risk of contamination, and they were way safer, virtually getting rid of the terrifying risk of an air embolism, which is where air from an empty glass bottle could get accidentally pushed into a patient's veins. So yeah, the next time you see a blood bag, just remember the incredible journey it represents. It's the story of going from doctors desperately trying to move blood from one arm to another before it turns solid, to this highly engineered system that keeps cells alive for 6 weeks with a precise chemical cocktail, all inside a purpose-built plastic container. And the story isn't over, not by a long shot. Right now, scientists are perfecting ways to freeze rare blood types for years, and they're even developing rejuvenation solutions that can bring expired red blood cells back to life. It really makes you wonder, doesn't it? What new miracles will be inside the blood bag of the future?